Can you see my screen?
Yes, Professor, we are able to see your screen.
So what I would like to discuss with you is the use of CAR T cells in order to develop immunotherapy for solid tumors. And I will divide my presentation essentially into three parts. First, I will describe some of the characteristics of the tumor antigen which we use as a target. Then I will give you some background about CAR T cells and why we utilize CAR T cells as an effector mechanism. And finally, I will show you some results which we have obtained with application of CAR T cell based immunotherapy for the treatment of solid tumors. Now, this work is preclinical, so it's essentially the treatment of malignant diseases is done in immunodeficient mice, but the results will then be utilized in order to translate the approach to a clinical setting if the results are positive. And I will mention to you already that this is already happening for one type of solid tumor. Now, what is going to the target, to the tumor antigen that we use as a target? It's B7H3, which is a type 1 transmembrane protein belonging to the B7 immune costimulatory and co-inhibitory family. There are two isoforms of B7H3 which are expressed on the membrane of malignant cells, and this is the structure of the two isoforms. As far as I know, it's not known which mechanism regulates the expression of the two isoforms and whether the balance between the two isoforms has a functional or clinical significance. Now, as far as the function of B7H3, it has not been completely clarified, but several lines of evidence would suggest that B7H3 has a negative impact on the immune response. And why do I make this statement? Well, because in several types of solid tumors, B7H3 expression has been found to be associated with a poor clinical course of the disease. So in a nutshell, we believe that B7H3 suppress the activity of T cells, which recognize tumor cells, and therefore provides tumor cells with an escape mechanism. But why do we use B7H3 as a target of CAR T cell immunotherapy? Well, in the following slide, I will summarize the reasons why B7H3 appears to be a very useful target for application of antibody-based immunotherapy. The first one is represented by the fact that B7H3 is expressed with high frequency, I would say, in all the tumors which have been tested so far. And as you can see in this line, which summarizes some of our work as well as information in the literature, in most of the tumors, the frequency of expression is at least 40–50%. Now, just I suspect that the antigen will be presented, will be expressed practically in all the type of tumors. And as an example to support my statement, I'll show now that uveal melanoma, which had not been tested before for expression of B7H3, when we finally had the opportunity to test the cell lines derived from uveal melanoma, we detected in all the types of in all the cell line which we tested. So that's why I suspected that B7H3 is practically expressed in all type of tumors. And in the case of uveal melanoma, we know already that B7H3 represents a useful target for CAR T-cell-based immunotherapy, as shown in these slides, which indicates that the B7H3 CAR T-cells can eliminate in an effective way, at least in vitro, uveal melanoma cells. So one reason why B7H3 is an attractive target is its expression with high frequency in many types of solid tumors. But another very important characteristics for antigens, for molecules which are evaluated, which are selected as target of immunotherapy, is what is the expression in normal tissues. Because clearly, if the antigen is expressed in many normal tissues, then any immunotherapeutic approach will cause many side effects and therefore will cause many problems from a clinical viewpoint. And so for that, we have investigated the question of the type of normal tissues which express B7H3. And as it's shown in the next slide, B7H3 has a very restricted expression in normal tissues. So actually, it's found only in adrenal glands. Adrenal glands is found on salivary glands, and that is found in the epithelial tissues of stomach, but is not detectable in the other types of normal tissues, therefore suggesting that when we apply immunotherapy targeting B7H3, we should not cause, or we should not cause many side effects in patients. And we have, of course, given the importance to know that there is no expression in normal tissues, we have performed several studies to prove these possibilities. And this is one example where we have used FACS analysis, and we have used IHC, immunohistochemical staining, in order to ask the question whether B7H3 is expressed for instance, in hepatocytes. And again, for comparison, we present the expression of B7H3 in a tumor of the liver, which is intracellular hepatic cholangiocarcinoma. Now, another important characteristics of B7H3 is that it's expressed not only on differentiated tumor cells, but it's expressed also on cancer-initiating cells. And the next slide shows that the cancer-initiating cells express a high level of B7-H3. So why I believe that this is important? Well, because according to the cancer stem cell theory, a therapy to be effective has to eliminate not only the cancer, differentiated cancer cells, but it has to eliminate also the cancer-initiating cells. Because if we don't destroy the cancer-initiating cells, well, those cells will generate both metastatic spread as well as they will generate recurrence of the disease. And according to the cancer stem cell theory, most of the defect, most of the failures of conventional therapy is that the therapies may be very effective in eliminating the differentiated cells, but are not effective in eliminating the cancer-initiating cells. And so just to show you in a graphic way what the cancer stem cell theory predicts is that most of the available therapies will destroy the differentiated cells, but will not eliminate, will not eradicate the cancer stem cells. And as a result, the cells which are left behind at one point after a period of dormancy, which can be short or long, they will begin to proliferate, will begin to spread they will begin to spread the metastasis, and they will cause recurrence of the disease. Now, on the other hand, if a therapy destroys the cancer-initiating cells, then this therapy has a very good chance of curing the patient who is being treated, because practically there will be no cells left following the therapy. Now, I'd like now to move to the second topic, which is what are CAR T cells, how do they work, and why are they an attractive effector mechanism. So CAR T cells are T cells which are genetically modified with a tumor antigen specific chimeric antigen receptor. And because of the genetic modification, T cells acquire specificity for the tumor cells, and therefore become able to recognize and to eliminate the malignant cells. So just to briefly summarize what the chimeric antigen receptor is, it consists of several domains. One domain is a recognition domain, which is usually a single chain derived from an antibody which recognizes the tumor antigen. So this recognition domain will give the specificity to the CAR T cells. And the single chain is linked to other domains which are mainly responsible for the activation and cytotoxic activity of T cells. So what happens when the receptor, when the recognition domain reacts, identify a tumor antigen, this will trigger several signals which will activate the T cells which have been genetically modified And as a result, the CAR T cells will acquire the ability to specifically eliminate the tumor cells which express the targeted tumor antigen. Now, before I go ahead, I want to stress that because of the limited time I have, I will not describe the slide or the data I show in details. However, please feel free to interrupt me or feel free to ask me a detailed question when we at the end of my presentation. Sorry. Now, let me move to the third part of my presentation, in which I will describe to you some of the neoplastic diseases which are targeting with B7H3 CAR T cells. And the main work I want to show to you is that related to the targeting of intrahepatic cholangiocarcinoma. Now, why did we select this type of malignancy as a target? Well, first, because it is the most frequent hepatic malignancy. But second, and most important, because surgery is the only strategy which might have a chance of success. But surgery usually can be applied only in a limited number of patients because the patients are diagnosed when the disease is already in an advanced stage and therefore has already metastasized. And the chemotherapy and radiotherapy, unfortunately, are not effective at all. So the first thing we did was to show that the B7H3 CAR T cells can recognize the intrahepatic cholangiocarcinoma cells, and both primary and metastatic, and that they can recognize not only the differentiated cancer cells, but also the cancer initiating cells for the reason I mentioned before. Now, just a second. So now we needed a model, to perform the in vivo studies, and in the early stage of our study, we identified that there was no available model, and so we decided to generate a model utilizing immunodeficient mice, because we are going to graft tumor cells, human tumor cells, in mice. Therefore, the mice have to be immunodeficient, because if they are immunocompetent, they will reject the tumor. And we, taking advantage of our expertise in surgical technique, we developed a model of intrahepatic cholangiocarcinoma, which requires the injection of the ICC cells to the immunodeficient mice. But this model is very useful, it's very effective, and also mimics the characteristics of the cholangiocarcinoma in patients, not only from a macroscopic viewpoint, but also in terms of the histopathological picture, as well as in terms of the expression of the antigen that we want to target. And the antigen is B7H3. And so once we had identified the model, then we had to perform the experiments by which we could identify what was the best, most effective strategy for the treatment of cholangiocarcinoma. And so the first experiment relied on comparing the efficacy of CAR T cells, which were injected either in the spleen or in the tumor, or were injected IV. And then the groups of mice were treated in this way. And of course, the cells were labeled with GFP so that we could follow the growth of the tumor by bioluminescence. And as you can see, in all the mice in which we injected the cholangiocarcinoma cells, we could detect a growth of the tumor. Then 2 weeks after the injection of the tumor, when there was development of the tumor in all the mice, we started the treatment utilizing the different routes I have indicated in my previous slide. And as you can see, the experiment obtained so far would suggest that the most effective route of administration of the CAR T cells is by intravenous injection, since in all the mice treated in this way, we could not detect the tumors, while the tumors was persisting in the mice injected either intratumorally or injected through the spleen. Then we, and this regression of the tumor is also parallel by the fact on the survival of mice. The mice which had been injected intravenously survived longer than the mice which had been injected through the other routes. And this mouse, which was lost for technical reasons. Now, then we ask the question related to the dose of CAR T cells, which is more effective in terms of controlling the tumor growth. And in the initial experiment, we compared the dose of 2.5 million CAR T cells per mouse and 5 million CAR T cells. And as shown in the next slide, we found that the dose of 5 million CAR T cells was most effective in eradicating the cholangiocarcinoma. Finally, we have investigated whether the CAR T cell, all these experiments, of course, address the question of control, elimination, eradication of primary tumor. But as I mentioned before, from a clinical viewpoint, it's important to know if we can control the metastatic spreading. And so, as shown in this slide, let me move to it. We performed an experiment in which we injected the tumor cells IV so that there was a metastatic implantation of the cells, and then we treated the tumor, the mice carrying the metastatic disease with B7H3 CAR T cells. And again, as you can see, in the mice treated with B7H3 CAR T cells, there was an excellent control of the tumor growth. Actually, there was an elimination of the metastatic lesion. Now, in the last few minutes, I have, for my presentation, I'd like to briefly show what we have done in 2 other types of tumors. One is a head and neck cancer. The other one is a triple negative breast cancer. And so we have asked the question whether we could increase the activity of CAR T cells expressing B7H3 by treatment with an HDAC inhibitor, SAR. And so as shown in this slide, the CAR T cells, which have been treated with SAR, appear to be more effective than the cells which have not been treated with SAR in controlling the growth of the tumor. So I hope that the results I have shown to you have convinced you, or have convinced you that B7H3 is an excellent target for implanting immunotherapy with CAR T cells, and the impressive result that we have obtained in the mouse model provide a good rationale to develop a to translate this strategy to a clinical setting. However, in order to do that, because of the concerns about the safety of treatment with CAR T cells, we are performing experiment in which we want to show that introduction of a suicide gene in CAR T cells provides us with the mechanism to eliminate the CAR T cells from the host in case there are unexpected toxicity problems. And so these are experiments we are performing at present. And finally, let me show to you, let me conclude my talks acknowledging the colleagues who have played a major role in the development of this project. Among them is my daughter, who is responsible for the cholangiocarcinoma program, and Dr. Dotti at the University of North Carolina, who has generated some of the CAR T cells utilizing the antibody that we have developed. Okay, thank you.